Welcome, Josephites. Thank you for joining us for the Jana Speaker Series session. Today, we have Josephite Dr. Kartik Chandran joining us for an AMA Ask Me Anything COVID-19 Safety session. My name is Anika, and I'm from the ISC 2009 batch St. Andrew's House, and I'll be your host for this session. A special thank you to the entire working committee of Josephites from across the US who have worked tirelessly on several initiatives like this speaker series. Logistics for today. The session will be recorded. All participants will be on mute, and you should turn off your video feed so we don't have any bandwidth issues. In case your video was set to turn on automatically when you joined, please turn it off. All participant questions will be sourced from our Jana LinkedIn page and the Jana website. Please post your questions there. The agenda for the next hour would be as follows. Gautam Majur will update us about Jana's initiatives. Francis Joseph, ICSE 2006, our MC, will introduce our speaker, Dr. Kartik Chandran, and moderate the AMA segment. This will be followed by closing comments, and we'll conclude with singing our school song. Thank you again for joining us. I'll now hand it over to Gautam.
Thanks, Anaga. Can you hear me?
Yes.
Great. All right. Hi all. This is Gautam Majur, batch of 89, Kartik Chandran and Rahul Ravid. Speakers in this series in the past were both my batchmates. Kartik, we sat in different benches, I in the last and you in the first, in case you don't remember. Let me start by congratulating and thanking the amazing young alumni working behind the scenes to reconnect and reinvigorate the OBA of North America, now called JANA. You can read more about these talented next generation alumni at the About US page on the JANA website, www.jana.co. Now for my usual disclaimer, if you hear some yelling and screaming in the background, moderator Giri, please do not unmute me. I have a 3-year-old and a 10-month-old, and my sense of work-life balance during these trying times have taken an unexpected meaning. The Jana Speaker Series started off with a bang with Rahul Dravid, and we have covered some excellent topics during 2020. You can listen to all these episodes at the Jana Connect on the website or Apple, Spotify, and Pandora. The team is getting ready to launch the Jana Mentoring Program and the Jana Entrepreneurship Program in December. Stay tuned for details yet to come. Jana has the much needed full support of the Bangalore Jesuit Education Society, the school's principal, and the OBA Bangalore leadership. We are really excited to announce that Jana has been newly incorporated as a Virginia nonprofit corporation on November 11th, 2020. We've been working very close with VJES and the OBA to establish the Jana Endowment to support the school and its programs for future generations, including student scholarships, midday meals, remedial classes, gratuity for staff, innovations lab, and several other initiatives. You can contribute one-time amounts to the general endowment, or if you would like to make a long-term commitment, you can sign up for a named endowment. Example, the John Doe endowment for the batch, John Doe endowment, or the batch of 1984 endowment. All contributions will be recognized on the Jana and the OBA website and the newsletters and other feeds. Details are available at the jana.co website. It will take a few months for Jana to get its US 501c3 tax deductible status. In the interim, we have arranged for you to receive tax deductible treatment for your 2020 contributions via a pass-through partnership arrangement with Learn for Life, a US 501c3 public charity which was established in 2012 by Thomas Takitela, batch of 77, to support education for disadvantaged children in rural India. All contributions made during 2020 will be tax deductible and hosted in separate accounts at Fidelity Investments and overseen by a third-party fiduciary CPA firm, as well as a JANA board. For named endowments, individuals will have the direct access to their own endowment accounts at Fidelity. Details about all this and more is available at the jana.co website. You can also email admin@jana.co for details on how to contribute. On behalf of the board and all the young alumni volunteers organizing this event, I want to thank you for joining the Q&A session with Kartik Chandran and Dr. Kartik Chandran for making himself available for the second time and for doing so at a short notice. In closing, I want to say that every endeavor has a champion Josephite in the driver's seat. This initiative does too. Thank you, Thomas Takitela, batch of 77, for your tireless effort and for kickstarting what Josephite should be indebted to for years to come. Thank you.
Okay, good morning, Josephites. Welcome back to the Jana Speaker Series. Today we're going to talk about COVID-19 and how to keep the virus out of our homes. We have Dr. Kartik Chandran, who we appreciate for coming back to answer some of our questions about the virus. So Dr. Chandran, can we start by telling us a little bit about yourself and what you do in your lab?
Yeah, so I'm a professor of microbiology and immunology at Albert Einstein College of Medicine in New York City. I have a research lab of about 17 students, postdoctoral fellows, faculty, and we study viruses. We study emerging viruses. We, of course, weren't studying COVID, but we were studying other types of viruses that are the kinds of viruses that we think can actually jump into people and cause disease, jump into people from animals. So our interests are to really understand how these viruses work, to try and identify the viruses that we think could be at risk of causing these sort of human transmissions and disease, and then to try and develop treatments for these kind of infections. So we collaborate a lot extensively with both the industry and government to take the basic research findings that we make and try and translate them into things like drugs that could be useful in the clinic.
Okay, well, you're definitely the right person to answer these questions we have here. So let's start with our questions. And if anyone here wants to post some questions, please do go to our website, jana.co, in the forum section and post your questions there, and we'll try to get through all the questions. Well, let's start with a person actually had a question about how do we know where the virus originated, and can we expect in the future, what can we expect in the future with such epidemics?
Well, I mean, the first question is very specific. The second question is very, very broad. So, yeah. I don't think, it's clear that the virus came from some kind of animal, most likely a bat. I think there's been some controversy over whether the virus was, jumped actually from nature, or it was a release from a laboratory or something. There's really no way. Well, I don't know, and I don't. Maybe that kind of information will come to light. There's no reason in principle it couldn't have come from been an accidental release from a laboratory, but there's also no reason in principle why it couldn't have emerged naturally from an animal. Yeah, I think my personal view is that regardless, this virus is a virus that's in nature. It wasn't genetically manipulated or anything like that, because we certainly don't see, the sort of molecular fingerprints of somebody messing with it. So, but of course, it's very hard to prove that something didn't happen. So, I think it's fair to say that a more full accounting of the events leading up to the initial outbreak in Wuhan need to be explained. Whether we'll actually get an explanation, that's not totally clear, but we'll have to
Sure.
see.
Yeah.
Um,
Yeah. Sorry. Go ahead.
Yeah, and then as far as the second question goes, I think we can expect lots and lots of things like this in the future. So just in the last 20 years, we've had 6 different outbreaks of novel coronaviruses in humans. 6 novel outbreaks, 3 in humans. So there's SARS, the original SARS, there's MERS, and there's SARS-2. But what's not really well known outside the virology community, and even actually they're not so well known because it really has to do with uh, uh, with a veterinary issue. So there have been 3 outbreaks of novel coronaviruses in pigs. Um, and these viruses in pigs actually can infect human cells in the lab. So in principle, there's no reason to think that such viruses couldn't jump from a domestic animal into humans. This has certainly happened many times before, in recent as well as in ancient memory, where viruses jump from animals to domesticated animals to people. So fact that in the last 20 years, we've had 6 different coronaviruses jump into either humans or livestock really tells you the risk that this group of viruses poses. There's hundreds of viruses like this in bats that have the potential to infect humans potentially without, as far as we can tell, very little adaptation required. And that's just one group of viruses. There are many groups of viruses that are totally different from coronavirus that also possess zoonotic risk, some of which you will be familiar, things like Ebola, but then lots of other more obscure viruses that we make it our business to study that most people haven't heard of. So,
Yeah.
and I think as human population grows, as climate change happens, as populations move, as, essentially, humans are kind of everywhere. Like we are, going into all these places that we've never been before in nature. So it puts animals under stress. It really narrows their habitats, the available habitats for them. All these things actually increase the probability of transmission to humans. So I would say that a lot of these outbreaks are, I don't think we should view them as acts of God in that human behavior and human civilization is actually driving these things. And so, there are changes we can make in the long term that reduce the risk of this happening, even at the same time that we actually prepare better for such
Yeah.
pandemics.
Yeah, that's a good explanation. Yeah, switching gears a bit, since we've had the virus for about most of this year, most of the questions are more towards what are the long-term effects of COVID? This is from a question from Dominic Shrestha, who comes from a hospitalist background. And he wants to know, post-COVID, what are the effects in humans? We probably don't know that, but has it been studied in rats or rabbits?
Yeah, I'm not really sure how useful those kind of models are. Probably studies in non-human primates might be useful, but um, it will take time to develop those kinds of models to actually to do. But I think there's an ongoing experiment, a huge experiment, which is significant proportion of the world's population is being infected. So we are already seeing a lot of long-term effects. Well, it hasn't been that long, but we're already seeing that there are these long haulers. There are these people that have cleared the virus, but seem to suffer some kind of long-term immune manifestations. We know this virus causes like a profound immune dysregulation so that people are almost developing like an autoimmune type of response to the infection. Not everybody, but some people. So the long-term consequences of that kind of autoimmune response is unclear. It may go away, but then it might also resurge at a later time due to some other sort of trigger. So, we could be creating a whole cohort of people that have some propensity to autoimmune disease. This is a kind of a speculative thing, but we don't really know yet, but it's possible. We know that there's going to be long-term, we know that lungs can heal post-COVID, but we also suspect that this is not going to be complete, especially in certain kinds of individuals. And so these individuals are probably going to suffer from reduced lung capacity, maybe permanently, maybe permanent lung scarring. They may be more susceptible to other kinds of infections down the road because of the immune inflammation caused damage that's already there. We know this virus has profound effects on the coagulation system, and directly and indirectly seems to affect both the heart and the brain. So, all of these things, so like if I use HIV, AIDS as an example, it took a long time for some of these more, what were thought of as more obscure effects, long-term effects to be recognized, but then eventually it became clear that they were quite profound and quite widespread. So one of them is these so-called HIV-associated neurological disorders, or HAND. And so these, in certain individuals with HIV, especially if it's been untreated, they can develop essentially early onset dementia because of inflammation in the brain caused by infiltration of immune cells into the brain, right? So we already know this virus can have neurological sequelae. So will it cause something like HAND? And are we going to see sort of a huge increase in these neurological disorders, especially early onset dementia? I think we're going to see this is going to be here with us for decades, because we're going to see a huge number of people with all kinds of side effects and all kinds of long-term sequelae that we're just starting to see now. So, and that's just the people that got COVID, right? I mean, so sort of, what always happens in these situations is that in this year, because of COVID, many people have not been able to get their medical needs met. So there are a lot of other people with chronic issues that, or even acute issues that, didn't go see a clinician, or didn't go to the hospital, didn't see the doctor, didn't have their regular visits with their primary care physician. Maybe they did telemedicine, but a lot of people probably didn't, right? So what are the long-term effects of on people that didn't have COVID because of their medical conditions being neglected, or because of preventive care not being done, or because of for screening for breast cancer not being done, or any of these things, right? So again, we're going to see a huge cost associated with COVID that's not actually directly due to COVID, but it's sort of a collateral effect. And this is just on the medical side. Of course, there are lots of other things.
Yeah, that's very true, actually. Also, we had another question. Let's say a person gets dengue or influenza. Will they be benign to the virus? Is there some kind of immunity you've seen? If you get one infection, it kind of protects you more?
No, I mean, these are very different viruses. So flu, dengue, those are very divergent. So there's no cross reactivity to the response. However, there are other circulating human coronaviruses. There's some very limited evidence that, we have constant exposure to these. So there might be some level of pre-existing immunity there. But there may also be things about how our immune response has developed to these other coronaviruses that may affect how we're responding to COVID. It's probably very dependent on the individual, their history, just chance. And then, what happens when, people are exposed to new coronaviruses, especially if we have a widespread vaccination campaign for COVID-19, if there's a slightly different coronavirus that emerges into human populations 10 years from now, what does that do in terms of people that have gotten the COVID vaccine? Will it protect them? Will it actually be counterproductive? So this is a big concern for viruses like dengue. I don't know if you guys remember, but when Zika happened, Zika is immunologically very closely related to dengue. So the concern was that, what would prior exposure to dengue do to people that had Zika? Now, in most cases, those fears were unfounded. Nothing terrible seemed to happen. But those kinds of concerns could really come to the fore for coronaviruses as well, especially if there are new coronaviruses that we become exposed to in the future. So, but we have to address the pandemic we have, right? We need to prepare for the future also, but we can't prepare for every eventuality, and we certainly can't predict what's going to happen. So I think we have to just go forward with the best thing that we can do right now, and then hope for the best for the future. But also, we need to do a lot to prepare for future outbreaks like this.
Yeah, that's very interesting you say that, because my next two questions were from Jyotirnath Ganguly and Thomas Takatala. They both want to know whether this virus mutates and whether it'll be like a flu shot where we get it every year. Um, so
Yeah.
so moving to the next section of like questions we got. This is more from a social impact rather than the science behind it. So this is a question from Jimmy Ankusaria, who said he read that beauty parlors and gyms account for only about 18% of the cases, and the remaining 82% cases from personal interaction between family and friends. So what is the purpose of shutting down the gyms, and which account for about 0.4% of the cases? And
so, I mean, I think the problem is these kind of numbers get thrown around, but they don't really mean anything because they're not really based on. So in the United States, our capacity to do any kind of contact tracing to really determine the source of people's exposure is very limited. So I don't know how trustworthy those kinds of numbers are. I will say this: there are certainly very strong, probably the strongest link to the rate of COVID disease and transmission is mobility, right? And there's actually a lot of good data for this using anonymized cell phone tracking, geo tracking data, using GPS data. So essentially, and this you can apply this at the national level, at the state level, at the regional level, and at the local level. Higher mobility is correlated with higher rates of infection, just in general. That mobility encapsulates, people going to work, people riding public transportation, people going to bars and gyms, and it's not one of these situations where people, yes, people are getting it by interacting with extended family or whatever friends. But where did that one person who infected everybody at home get the infection, right? Like he or she may have picked it up at a bar or a gym or at the grocery store. I don't know. But the thing is that it's really about networks of connectivity here, networks of interactions. And from a network perspective, mobility seems to be the number one sort of number one determinant of the hospitalizations, for example. So actually, New York Times had this really nice graphic that they published like yesterday, where they showed that in the U.S., when you look at different states, you can calculate like essentially a mobility index, which builds in things like lockdowns, mask mandates, and all these things. So like a sort of an overall number that tells you something about how the state responded to limit transmission by limiting mobility, closing bars and restaurants, having a mask mandate, and then the current rate of hospitalization. It's almost like a perfectly linear correlation. So the more you kind of limited people's mobility, the lower the rates of hospitalization currently. So like South Dakota, like North Dakota is like the worst, right? And they're like all the way up on the, they've done nothing, and they have the highest, they're literally the single jurisdiction like in the world with the highest rate of hospitalizations per capita right now, because they've literally done nothing. In fact, they've done the opposite of nothing, right? So they've encouraged people to go out and make merry and not wear masks and not give a crap. So so we know, I mean, if you look at New York City and the Northeast in general, we know that when we lock down, I'm not saying we should lock down all the time. I'm not saying, but we know that when we restricted mobility, when we made people wear masks, when we closed indoor dining and bars, inside sort of having drinks in bars, we were able to completely reverse, the spread and flatten the infection. And for all of summer, we were like below 1%, right? Now it's starting to go up, in part because people are getting tired, because we're opening more things, and because more people are coming from neighboring, states and so on that are bringing the infection in. So, we know that restricting mobility works. Now, obviously, there are trade-offs, but the notion that, bars and restaurants and indoor dining aren't contributing to spread, I wouldn't agree with that. I think there's also independent, I mean, we don't have time, we could talk about this for hours, but there's independent data directly relating many. The other thing to remember is that spread is like, there are certain individuals, then certain situations where you get lots of spread, right? Like one person seems to infect lots and lots of people, right? So the household attack rate, meaning that if I were infected and then I came home, the chance of my family member, one of my family members getting it from me is actually only 20%, even though we're together all the time, we're not wearing masks in the house, right? But, in another situation, when you have a crowd of people, one person can give it to 100 people, right? So it really has to do with viral load and, how much virus is being expelled and what is the nature of the social interaction. If it's a lot of talking and singing and dancing, especially without masks, then it's almost certain that, and the more people you crowd into one spot, the more likely it is that one of them is going to be one of these kind of super spreaders. So it's really a statistical probabilistic phenomenon, which is related to mobility, and it's also related to the number of people you crowd into an indoor space where the virus can hang around in the air for a long time, especially if they're talking and dancing and they're not wearing masks and they're even if they're wearing masks, they pull their masks on to drink and eat. And every time you eat in a restaurant, I mean, it's kind of disgusting, but there's actually saliva, there's mucus everywhere, right? I mean, like sharing a cocktail with someone is like the equivalent of giving them a French kiss, like literally. So, so these things do spread the virus. And so the notion that we should just open all the bars and restaurants, I mean, if we do that, we'll have North Dakota basically. So,
Okay.
yeah.
Yeah, got it. So human beings being very social beings, they need, and the virus has been around for some time, so people are definitely fatigued. So what are like the precautions you can take if you want to go out? So how effective are these
I would say
masks?
this, right? The end is near.
Okay.
Right, we have at least 2 fantastic vaccines, but more coming along. It's going to take some time to roll them out, but eventually we are going to have vaccines, and many people will be able to get them, especially people at higher risk. Um, Herd immunity is not a solution, right? The only kind of herd immunity that's going to work is one that we get through vaccination. Naturally acquired herd immunity means we're basically saying we accept, several million deaths, right? Like it's not acceptable, at least to me. I think it will destroy our medical. It will completely gut our hospital system. I mean, you're saying everybody's tired and blah blah, but I think what we really should be thinking about are the doctors and the nurses that are in the hospitals that are seeing this every day, that are putting people in body bags and in morgue trucks, right? Like, think about what it's like for them, right? I mean, so we have to, yeah, okay, it sucks, right? We don't get Thanksgiving this year. Maybe we don't get Christmas, but I think that's really kind of, while unfortunate, it's a sacrifice that we have to make you know, so that our seniors don't have to die, alone in nursing homes, and so that our the people that are actually taking care of us in hospitals, who are already running, doing incredibly long shifts, who are already exhausted, who are already mentally exhausted, from seeing so much death, right, and feeling hopeless about it. We need to care about them because they are at the end of their ropes, right? So if we don't behave responsibly as a community, then I think, eventually these people are going to throw up their hands and say, forget this. Like, why am I doing this? I'm just going to go home and take care of my own family. And so we have to be mindful about that. As much as everybody's fatigued, we have to just accept the situation we're in and act accordingly. And, we'll have a great Christmas and Thanksgiving next year. We'll have a great summer vacation if we all behave, do what we should do. If we don't, then this is going to keep going on. So, and it's going to take that much longer. And it's hard to get groups of people to do things, especially in the face of active opposition and disinformation from the highest levels in government. So that makes it much, much worse. But, that's the perspective that I have. I haven't eaten at a restaurant, even outdoor dining. I don't, of course, haven't gone in a bar. And, I get takeout, I get food delivered to my house to try and support restaurants and things like that. But, I'm not going anywhere for Thanksgiving, and I don't plan to go anywhere for the Christmas holidays. Maybe or at least I don't plan to get together with other people. And I'm accepting it as something that I have to do for the greater good. And I think that's just, but I think at least we can say that the end of the tunnel, the light is at the end of the tunnel is visible. But you know, so now is not the time to sort of, say, screw it, I'm just going to go do whatever because it's the holidays, right? Because it's just going to prolong the situation, right? If we all act responsibly, then we can actually bring this to a conclusion in 2021. Otherwise, I don't know how long it's going to go.
Yeah, you mentioned about takeout. So do we have to take any precautions about bringing food from outside? Is that a low probability?
It's a low probability, but I think if you're worried about it, there are a few things you can do. So one is, like if you're bringing takeout into your house, if there is contamination, especially if it's cooked food, which will destroy the virus. The real issue is outside of the containers, right? That maybe if somebody was handling it that was infected, that there could be virus on there. So, of course, the easiest thing to do is just to transfer the food out of your takeout containers and put them in your own containers. Dispose of the takeout containers, and then wash your hands with soap, and then eat your food, and then that's fine. If you want to go one step further, you could wipe down the outside of the containers with alcohol or something. That takes, if you have alcohol in the house, which everybody does, then that takes an extra second. So either of those things. So it's a low probability event to get infected like that. But on the other hand, it also only takes a couple of minutes to address that risk, even if it's a low risk. So that's sort of that's what we've been doing.
Okay, that definitely makes sense. So the infection rates in like countries like Southeast Asia, South America have been dropping. Do you have any sense why that's so? Like even in India, in certain areas.
I have absolutely no confidence that that's actually true in India because they basically just stopped testing. So I'm not convinced. I mean, given the state of our medical system, I'm not convinced that you can actually believe any of the numbers coming out of India. I'm just being honest because, I mean, I know people, obviously I know lots of people in India, and I know people that work in hospitals. I can tell you that all of a sudden it's become difficult to get tests. It's not clear that data are really being kept properly. I just don't know that we're tracking the information in a way that you can reliably say the cases are dropping. And I think exactly the same is true of most countries in South America, except maybe Chile or something, where they're sort of better at this. So yeah, I mean, I guess I would dispute the premise of your question, which is that the cases are dropping.
Sure.
Even with assigned deaths, right? Like do we really have an accurate count of how many people in India died of COVID? Because, if they weren't tested for COVID, right, then what are we going to do? Test dead bodies? We're not even testing live people. So, I'm not convinced that you can even really trust the number of deaths due to COVID that's being reported in many places.
Could you have a correlation between like number of beds occupied in a hospital with COVID to maybe the number of COVID in that area? Because
you might.
those numbers have decreased.
Well, I mean, so if that's true, then maybe that there are fewer cases. The government did do some things to reduce mobility at various points. But, I think, remember that these are all lagging indicators, hospitalizations, deaths. So, let's see what happens after Diwali, when, as far as I can tell, there were no real restrictions. It's the thing with India, like in the US, is it's very patchwork, right? Like some places, like I'm told that it was, there were there's a lot less activity. Other places, it was just like business as usual. So we should start to see discrepancies in local sort of rates of hospitalization. Yeah.
Got it.
Again, how many people go to the hospital? I mean, how many people die at home? I mean, lots of people have died at home here too, because, we have such a terrible, balkanized healthcare system, and so many people don't have insurance, so maybe those people just don't even go to the hospital.
Sure.
Maybe they wait too long because they don't want to go to the hospital, and then by the time they go, it's too late, or they even die at home. So
Yeah,
I'm just saying, take all these numbers with a grain of salt. Yeah.
understood, understood. Yeah, so you touched about testing, and so there are a bunch of different tests out there, some rapid tests, which you get results very quickly. So what's your take on getting a test done, and what maybe the steps we need to do? Let's say we think we got infected yesterday, how long we should wait to get the test and things like that.
Yeah. So, okay, let's talk about the standard sort of test right now. That's really the best test we have, the gold standard, which is the qPCR, the Rtpcr test. So this is a thing where you do a nasal swab, or you can even use saliva. People are using saliva now. And you're measuring the amount of viral nucleic acid, RNA, in the mucus or the saliva. And this test is very, so if you got infected yesterday and you got this test today, you would probably be negative. So I would say, on average, it takes about 3 to 5 days to become positive in this test, because there is that bit of incubation time. Now, is it 100% at 5 days? No. I mean, some people don't really start to show viral RNA in their nasal passages until like 10 days. So it's a moving target, right? But in general, statistically, if you do a test on day 5, and that's why the quarantine is set at 2 weeks. So I would say, if you were, for sure you were in close contact with somebody that was exposed, then basically, you should quarantine. You should basically plan on getting 2 COVID tests that are negative. So I would say test on day 5 and then test on like, day 10 or something. And if those 2 both come back negative, then you're probably fine. But if you test too early, it won't be useful. And if you only do 1 test, then it's hit or miss, because even if you test on day 5, it's possible that you might miss it. Although in general, I think if you test on day 5 and you have, the other caveat, of course, is symptoms, right? So all of this assumes that you're completely asymptomatic, right? Obviously, if you have symptoms, you should quarantine for 2 weeks. But if you don't have symptoms, then I think getting a test on day 5 is probably good enough, because the state of things right now is that it will take you 3 or 4 days to get your test back anyway. So it's gonna be sort of, you're gonna have quarantine for like 7 to 10 days, and I think that's probably, if you want to be really safe, I would say 2 weeks, and then get a second test towards the end of that 2-week period. And if those both come back negative, then you're fine. The rapid tests, the problem, okay, so I think rapid tests definitely have a place and a value. But right now, rapid tests, the problem with rapid tests in general is that they're in this uncanny valley. They're too expensive, and they're not that, so what if if we had a rapid test that was like these glucose strips, that you can buy at CVS, and you can just, it wouldn't be blood. It would be like like a nasal thing or a bit of spit or something. And you put it in a reader and you get a result. And you could buy a pack of 50 strips for like $10. Then you could test yourself every day, right? And then that's really what you need to do with those rapid tests, because they have a high, there's this concept called the positive predictive value, right? Like, so if I get a positive test, how predictive is that I actually have COVID versus just being a false positive? And that's very dependent on the overall positivity, right? Like, the lower the overall positivity in my neighborhood, the sort of less that positive predictive value is, right? So just to give you an example, if the overall rate is 3% in the community, the positive predictive value of most of these rapid tests is only 50%. So it's like a coin flip whether you got it or not. However, right, that's one test. If you test yourself 3 days in a row and it comes back positive every time, right, then that's a totally different situation, because the probability is basically multiple, right? And the probability that you would be positive 3 days in a row is almost 0. So But unfortunately, the rapid tests that we have right now, the rapid tests that we need that would allow you to do this are just starting to come on the market. And even the one that, like, it's, you know what I mean? So that's the problem, right? Like the rapid tests we have right now are just not good enough, and not available enough to be able to use in this way. A lot of people are using rapid tests sort of as an adjunct to, because it's a lot faster to get the results of a rapid test, right? So they're sort of saying, well, okay, get a rapid test. If you get a positive result, then get a PCR to confirm it, the confirmatory test. If you get a negative test, then you're fine. But the reality is, again, the question is a single rapid test that's negative. I'm not sure what that means. You really need to be able to get it multiple times, multiple days in a row. So I think rapid tests are ultimately the way to go as the first line of defense. But we still don't have the rapid test available at the kind of scale and cost that makes it repeated serial testing with a possibility for individuals. And ideally, you have to be able to do this at home. Like you shouldn't have to go to the clinic or something, or need a prescription. You need to be able to buy it over the counter. So unless and until that happens, I don't think rapid testing is really going to be that useful. It's useful on the aggregate level to do like to look at surveys of prevalence on a population level is much more useful right now, the kind of test that we have, right? But for an individual who's asking, well, I need to go to a wedding, and am I positive or negative? Like, if I could test myself for 5 days prior to the event every day and I was negative all 5 days, I could feel pretty confident about it, right? Now, of course, I might have gotten infected that 5th day, and so that's the problem, right? There's always a lag time. So you can't be 100% sure about it, but you can at least, do the best you can. And that's why we're saying ultimately, don't go to a wedding with 50 people, and nobody should be holding such in-person events right now until we can get enough people vaccinated, because it's not possible to keep people safe.
Yeah.
There's no magic bullet right now for this.
Yeah. Giridhar Naik, I hope that answered your question. And so you did mention the light is almost here. I mean, we're almost at the end of the tunnel. So we have a few questions on vaccines. And there are different kind of vaccines coming out, like mRNA versus the classical type of vaccine. What are your thoughts on either? And would you prefer one type of vaccine to the other?
I think the mRNA vaccines are very new, but the data that's coming out of the two companies and there are actually other companies too that have mRNA vaccines, looks really promising. They look very safe on now the thing is that ultimately you would have to roll these out to lots of people in order to, some things might crop up that we aren't anticipating, but at least in the sort of the phase 3, phase 1, phase 2, phase 3 work that's done so far, there don't really seem to be any significant sort of acute adverse effects. Nor would you really think that in the situation with an mRNA vaccine that you would really anticipate anything. I was more worried about efficacy, but the efficacy data look good so far. So I mean, I'm very like, I feel much better about the RNA vaccines than I did a few months ago. The other sorts of more traditional vaccines, insofar as you can call them traditional, so there are these, I think there was a question about the Oxford vaccine. So that is like a adenovirus vector vaccine. There are other such types of experimental vaccines out there. And, those also probably will be just fine. I have no idea how good the efficacy is. That's not totally clear yet for some of those. But ultimately, there are two sort of purposes to getting a vaccine. One is for the individual to protect them. And then the second is to epidemiologically to reduce spread. And so in the first situation, obviously you want the vaccine to be as effective as possible, because that, a 95% effective vaccine is really, really good. A 70% effective vaccine is like, but it's better than nothing. It's better than a coin toss, I guess, but it's better than nothing. But on the population level, even a 70% effective vaccine will have a huge effect on reducing spread. So there's really two games here. There's a game for the individual and a game for the population, just as it's been all the way through with everything else with this virus. So, so I think, what's going to end up happening realistically is that we're going to have all these different kinds of vaccines become available in different jurisdictions, and then people will just need to go get them. We should have the kind of safety data, in the months to come that will make people feel comfortable. But at the moment, I'm not, there's really nothing about these vaccines that's particularly at issue. They're all, the mRNA vaccines are much more sort of innovative, but, so far they're, people have been experimenting with them in animals for some time and haven't really noticed anything untoward. So so if I had a choice right now, I would probably get the Pfizer shot or the Moderna shot if it was available, right? And so again, both of those are 2 shots, right? So there's a prime and a boost. So every vaccine will have to be evaluated on its own terms. But again, as these get rolled out, we'll start to collect more data. All these companies will keep data on, on side effects and efficacy and things like that. So.
Got it. Yeah, so let's say we start getting vaccinated, and what level of vaccination should be prevalent so that the virus disappears? Let's say 70%, 80%. How many people need the vaccine for the virus?
I mean, it's hard to know for sure, but I would say upward of 75, 80%.
Okay. And you mentioned there's probably no possibility. Going towards herd immunity is not the way to solve this problem.
The way we go towards herd immunity is by vaccination.
Okay.
We don't get to herd immunity by letting people get infected and letting them die. I just think that's immoral. So.
Got it.
Although lots of people seem to think it's just fine, but I think you'd be hard pressed to find a single like a medical doctor or nurse who would think it's okay.
So the Serum Institute in India is already set up to give about 100 million doses of the Oxford vaccine. Do you have any thoughts on like the studies they've published?
I think it looks perfectly fine. I think it's a good vaccine. It should be fine. That should work pretty well.
So Pfizer versus Oxford, there's probably.
I think it's really about access and practicality, right? So you may want, if you're in India, you may want the Pfizer vaccine, but you may not get, if it's not available, then what are you gonna do? I would get the vaccine that's available, and if that's the Oxford vaccine, then it's fine. I mean, the Oxford vaccine there's absolutely no reason to have any. At least from the data I've seen so far, it looks great in monkeys. I think there's already some preliminary data coming out of people. It looks safe. Anytime you do any kind of vaccine trials, you're always going to have some people having some kind of adverse effect. If the trials are large enough, it's not entirely clear if that's because of the vaccine or just by chance. So if you take a random group of 10,000 people, at any given time, there are 3 people that are going to be having heart attacks, right? So it doesn't mean that the vaccine did it, right? So you have to also read the headlines carefully and not sort of, the news media often hype some of these things as, Oxford trial stopped. Well, that's just a responsible thing. Every one of these clinical trials has something called a data safety monitoring board, which is an independent group of people that look at all the data, and basically they're the ones that decide if a trial can continue or not, right? They can stop a trial because there's a problem. That stoppage can be temporary or it can be permanent. They can also stop a trial because the vaccine works so well that it would be unethical to continue to essentially do a trial when the outcome is already known. And so it would be unethical at that point to continue to put people in the placebo group, right? When they really should be getting the vaccine. So, so that hasn't happened. And some of these vaccine trials have been temporarily stopped to investigate certain adverse event reports. But, I haven't seen anything that really rings, an alarm in terms of safety. But of course, the more people get it, the more, if there are some very rare things, we'll start to see it, right? So a lot of times it's not just the vaccine, it's the way the vaccine sort of synergizes with the particular kind of individual. So, it might be 1 in 10,000 or something, like, you're not going to see it till you test like 100,000 people, then you might get 1 or 2 cases, right? So it's the scale of the testing, and so as these vaccines really get, I mean, if Serum Institute of India is really going to vaccinate 100 million people, then if there's any a lot of crazy things will happen, because if you take a random group of 100,000 people, a lot of crazy things are going to happen to some people, right? The challenge will be to determine if it has anything to do with the vaccine or not. But of course, with that many people and with the rate of COVID spread, we'll be able to tell very quickly whether the vaccine works or not. That's the other good thing, right? If the rate of the virus is very low, then it becomes very difficult to establish the efficacy of the vaccine, because you're basically waiting for people to randomly get infected in your trial. And if only like 10 people get infected, then there's not enough good data to really statistically tell if the vaccine did anything or not. But if the way things are going right now, lots and lots of people, that's why Moderna and Pfizer were able to determine so quickly that their vaccines were highly efficacious, because so many people were getting infected in their control group that, and then of course they were able to see that there were very few infections in the experimental group, right?
Got
So,
it. Yeah, so we have some questions coming in right now, actually, and one is along these lines where, so since the mRNA vaccine intervenes directly with the genetic material of the patient, what are your thoughts on this, and is it a valid concern?
I think it's so it doesn't actually really intervene directly with the genetic material of the patient. So mRNA, your DNA is in the nucleus of the cell, and mRNA is in the cytoplasm. Basically, the way the vaccine works is that it's actually not just naked RNA. Naked RNA is not stable. It would get destroyed in seconds. So the RNA is actually encapsulated, or it's wrapped in a kind of a greasy shell. It's called a lipid nanoparticle, sort of like a membrane, and that's what is given, and it basically goes into the muscle and it's taken up by cells, and then a few cells in the muscle, right, actually take up the RNA, and that RNA reprograms the cell to make a protein, which is actually the vaccine. Okay, so it's normally what would happen is that the protein would be manufactured and you would get the protein, right? Here they've just short-circuited that by giving you the messenger RNA, which goes into cells and makes them produce the vaccine in your body, right? The mRNA goes into the cytoplasm in a few cells and it produces the protein. The mRNA doesn't go into the nucleus, right? And so it's not going to interfere with your DNA. There are millions of mRNAs in your cell at any time, in all your cells, and these mRNAs are just a few more, in your cell. So it's sort of like adding a few more drops of water in a bucket. So the RNA itself is not really a concern. Also, it's temporary because the mRNA is not going to stay there forever. It's going to stay there for like a week or something, and then produce a protein, and then it's just going to disappear. It's going to get degraded. It's not infinitely stable. In the cells in your body, mRNAs are constantly being degraded, turned over. So then that will happen, and that's why you need another dose of the mRNA to get another shot of protein. So at least in principle, there's absolutely no reason to worry that the mRNA is going to do something to your genetic material. I think that's not really a concern. If there are concerns about the vaccine, they really have to do with formulation. So what else is in the vaccine? So a lot of these vaccines contain an adjuvant that stimulates the immune response. 9 times out of 10, it's the adjuvant that causes trouble, not the vaccine itself. So, so how are these vaccines adjuvanted? How are they formulated? What else is in there? So those are really the issues. It's usually not the active ingredient that's the problem. It's all the other crap that's in there that goes in there to make sure the vaccine works. But all of that's being tested. So there's no real reason to be concerned about the RNA versus any other kind of. And similarly, with the Oxford vaccine, that's an adenoviral vector. So you're actually getting infected with a virus. But that virus is being engineered to be harmless, and it also goes into cells and dumps its DNA. So, in that case, actually, the DNA, goes into the nucleus, right? So it's actually even closer to your actual DNA in your cells than the RNA, right? But it's the same idea. The DNA programs the cell to produce the vaccine. It's just that the way that reprogramming is happening is a little bit different. In the case of Mrna and Pfizer, it's the mRNA that's reprogramming the cell to make the vaccine. In the case of the Oxford and some of the other vaccines, the Russian vaccine, it's the adenoviral vector, which is a virus that it's basically been stripped of all the other things. So it's got only like these essential components. It's basically a delivery vehicle. And so this delivery vehicle goes in and puts the DNA of the vaccine into the cells, and then those cells make the protein from the DNA. And they also make RNA. So the DNA goes into RNA, and then the RNA made into RNA, and the RNA is made into proteins. But there also, the DNA doesn't last forever. It eventually goes away. So, in all these cases, in all these vaccines, some kind of genetic material is ultimately being delivered to the cells to produce the vaccine. It's just the mechanism and the way that it's done is different in each case. But those types of systems have been used extensively and are been tested. So there's really no real safety hazard associated with that. Right. Also, these things will not. The real risk would be that if they were inserting into your DNA in your cells, and there's really no reason to believe that would happen.
Okay, makes sense. So maybe let's end on maybe a slightly philosophical question where we see vaccines as a solution to these problems, but maybe we should stop looking at, we should look at preventing this from ever happening. So what maybe things we can think about to prevent these viruses from mutating to humans and things like that?
Well, I mean, there's a lot of things that need to be done. So one is so you can put it all under this broad umbrella of preparedness, and preparedness has lots of different components. So the one thing is to reduce human impact on the natural environment. This is the root cause of all of these things, ultimately. So, and that includes climate change. It includes deforestation. It includes human encroachment into natural areas that stresses out all these, creatures that live there. It includes reducing things like bushmeat practices, wet markets, things like that. But also realizing that people have to live and people have to eat. So, a mandate without some other sort of accommodation for these people is not, people are hunting bushmeat for reasons, right? So we need to, just like how in recent years we've been really successful to reduce poaching, not just by killing poachers or arresting them or whatever, but also understanding that, how to support their community so that they don't need to poach, right? Like making them a part of the solution. So I think those kind of more creative solutions to reduce exploitation of natural habitats by humans, I mean, that means we have to change our entire, culture and how our global economy is structured. And so these are sort of like the big challenges if you really want to reduce the risk of these kind of zoonotic transmissions. Then, from the perspective of like preparedness of, we need to actually invest money and time and resources in developing countermeasures, whether they're vaccines and therapeutics, both, against lots of different potential zoonotic threats that we know about, including some future proofing so that, something that we're starting to do and think about in other people is like, how do we prepare for a future coronavirus outbreak, right? Like the things that are going to work for COVID, like these vaccines, are not going to work for some new coronavirus. So we need to build in some robustness to these vaccines and drugs so that they can work against multiple coronaviruses, not just one or two of them, right? So it's sort of like, having a bigger umbrella of protection. And this is just for coronas, there's lots of other things. So we need, for each one of these groups of agents, we need to have some umbrella of protection. That means you have to invest money in research. We have to invest money in, we need some sort of process that will allow us to very quickly, with government and private investment, bring these types of products to a point where they can be very quickly deployed. So the traditional approach of FDA licensure is not going to work because there's not enough, it's too expensive, it takes too long, and we don't really need it, right? Like none of these vaccines and drugs are going to be licensed by the FDA. They're all operating under emergency use authorization, right? So, we just need to get to the point where we can do a phase 1 clinical trial do the preclinical studies that get you to that point, do a phase 1 clinical trial, which just shows that it's safe, and then do some very limited efficacy work, maybe just in animals, and then you have something and you park it. And so if there is an outbreak, then you've already got something that you can kind of scale up. Now, coming to that, we need really like a coordinated plan. So the way the US has responded to COVID-19 has been disgraceful, right? So it's marred by willful denial of what's happening, combined with incompetence at every level, right? Like if you look at certain jurisdictions, like the Northeast, they've done better. But on the whole, it's terrible. And so, the US doesn't like to do things in a coordinated way because of blah, blah. But, but pandemic response is the one sort of thing that has to be coordinated, at all levels, including federal, state, and regional and local. It's just like any other kind of disaster response, right? So we need like systems in place to really to roll things out in a systematic way, right? Like, how are we going to roll out all these vaccines and drugs? Like, who's going to manufacture them? Maybe we have to like have a plan in place where we actually pay companies, when there is no pandemic, sort of like a retainer, right? So that if there is a pandemic, they will immediately divert all their production to making a vaccine or something, because they have other products they have to make, right? So I think it's about diversifying our supply chains and making them more robust. It's about looking at those vulnerabilities and addressing them. Stockpiling, obvious things like PPE and medical supplies and, masks and blah, blah, blah. And that sort of all goes along with this preparedness piece. I think we also need to have a robust public health response. That means we need to invest. We've disinvested in the CDC and organizations like that are really central to coordinating these kind of responses. We need like kind of very strong support of the epidemiological framework that exists, that has existed in the past, that has been so important. And that's just not, complaining that the CDC is not doing its job, but they can't do their job if their budget's being cut by 30% and they're being constantly interfered with by the politicians. So, we need to put people in charge of these things that can just, can like essentially execute a formulated plan and have the resources to do it without, politicians getting in the way constantly, right? So there's many different aspects to this, and I think we have a lot of soul searching to do, like not just the US, but just globally in terms of how to address this, because this is going to happen again, and it's probably going to happen sooner than we think.
Yeah. With that, thank you for all these insightful answers. I thank all the Josephites who posted these questions. I understand that few of the questions we haven't been able to answer, but we'll try to get Dr. Kartik Chandran to answer those on the forum, jana.co/forum. You can see the screen share right now. Please post any questions you might have there, and we'll try to answer them in the forum. And with that, we'd like to end this talk. Thank you, Dr. Kartik Chandran. It was great listening to you.